If stem cells are going to be the way we treat disease and injury, then banking your stem cells will become the most important investment you can make for your future. To bank or not to bank? That is the question.
In the not-too-distant future, a revolution is brewing, one that will change the very fabric of our existence. It's a tale of cells that hold the key to unlocking the mysteries of life itself. Let us venture into the unknown, for in this age of stem cells, anything is possible.
I've seen some beautiful flowers.
Welcome back.
Happy spring.
It is spring and it is very loud around here. We are well underway, our renovation project, improvement project, and please excuse the noise. We'll do our best to get through this. Today's topic is adult stem cells. Adult stem cell banking is what we're going to be tackling. Most of you are probably familiar with newborn stem cell banking. So when you're pregnant, about to give birth, you'll be often pitched, do you want to bank your baby's stem cells? Now's a great time. Now's the only time to bank, is what they'll tell you. We have a great episode focused on that specifically. Taylor, please add the link somewhere here. If you want to do a deep dive on newborn stem cell banking, please watch that. Today, we're going to be talking about adults. Now, going back to the babies for a second, last month, did you hear about the lawsuit?
I did. I will confess, I did. And I'm trying to decide if it was unexpected or not. I know that we'd predicted elsewhere that we thought we'd see more legal conversations around regenerative medicine practices, and I think it's been well known in our industry for some time that not everybody is perhaps acting or communicating the way that we think they should. One would hope that they would. Perhaps a consumer or client hopes that they would. But I'm going to say unexpected because I was surprised when the news crossed my desk that.
So last month in February, Texas Attorney General Ken Paxton sued Cooper Surgical, which is the parent company of CBR. This company, formerly known as Cord Blood Registry, was acquired by Cooper, and they are the biggest. I think they hold over 1 million units of cord blood that's privately stored, but they are the biggest cord blood bank. And we call them cord blood bank because that's what they are. Cord tissue. They also provide tissue banking in the United States. They also serve like 100 countries, so they're pretty much a global name at this point. But they're being sued. And they're being sued for, the allegations are misleading marketing, claims that stem cells could treat many diseases, and selling biological insurance. Now, Texas is actually the second state that's suing Cooper Surgical. The first state is Arizona. That happened in March of 2025. And Arizona's Attorney General did the same thing, deceptive marketing, failure to properly disclose sample quality issues, right? So this becomes an area where, should you know the health of what you're banking? So let's say you're paying for a banking service. Should you know upfront whether this stored tissue or blood is any good, right? Yeah, so that quality. And misleading claims about physician endorsement. So that Arizona case is still going on. The judge did refuse to dismiss it, and that case is proceeding. And there are also other class action lawsuits. In 2024, there was one as well related to false advertising about the usefulness of cord blood and the failure to disclose very low probability for autologous use. I was so surprised in the Texas lawsuit, they said that the chance is near to zero. And how near to zero? Again, watch the episode from before, because we actually did the math. And the math doesn't math, right? When you're talking about how these banked sources actually convert to treatments later on. So, in today's topic about adult stem cell banking, we want to tie in or use as a good ground for what's happened in history. So the cord blood industry, which started about 20, 25 years ago, is a good foreshadowing of what could happen or what would happen in the adult stem cell banking industry. And we'll see how that, what we've learned. Let's start there. What are we learning from what was done in newborns?
When I think about the experiences of the families who have, who are part of this newborn stem cell making industry, I think the theme that comes up, and it comes up in these lawsuits, at least one of the themes is, yes, we need consumer protection laws and so on and so forth, but it's also extraordinarily important, like any other purchase, for a consumer to be well educated and as well informed as they can possibly be about the decision that they're making. And I think that's what gave rise to today's conversational topic is what is stem cell banking? We kind of throw it around and we talk about it, but let's actually pause and explain what we're doing, why, what we're not doing, why, how an individual family or a person in need can make a decision about what's right for them. And again, why or why not?
Yeah. Well, maybe to start, let's define what is stem cell banking. Again, there are different processes that can take place, and different people can market their services as stem cell banking, but it may not necessarily be the definition of stem cell banking. So all in all, I think what we can agree on is stem cell banking is collecting and cryopreserving, because you got to preserve them some way, and cryopreservation in really cold temperatures is the way you preserve cells. So collecting and cryopreserving stem cells for possible future use. That's banking. When you bank money at the bank, we bank for future use. There's also this notion of, well, it's not a notion, it's the act of withdrawing. Depositing and withdrawing. In this case, you deposit once and you should have the ability to withdraw. So you should definitely understand what the withdrawal terms are and what you're withdrawing. So what you're depositing is also equally important, because you deposit this once. So you have to know, is what I'm depositing worth withdrawing in the future? And a lot of people don't think about banking in that way. They think about it like what the language that's been used previously, biological insurance, like just in case. Well, this is no different than buying an insurance policy without knowing whether they'll pay at the end of the day, right? And there's lots of situations where they'll say, oh, I'm sorry, we just can't, for whatever reason. And I think in the cord blood world, there have been many instances where patients have tried to access cells, whether in the context of a trial, only to find out that there were quality issues. That's why these lawsuits are taking place, because they find out later on, when it's time to use them, that what they banked was not usable, probably from the beginning.
Yes.
Yeah.
Yeah. Well, I think there's a scientific learning curve there as well, because and we're going to talk a little bit later, I hope, about types of stem cell banking. But, if you're banking a cellular product versus a tissue, right? There's a longevity window on that as well, and so that's another base level of knowledge that I think if people had this information, in a readily understandable format, which is hopefully what we're doing today, right? It can help them make better decisions and decisions that are more clearly and realistically communicated. I mean, like what we see in the newborn banking scenarios that resulted in these lawsuits is commercial companies arguably preying upon extremely vulnerable people about to have babies, so emotions are high, perhaps their health concerns, to make large dollar purchases that they may or may not understand. And again, without saying that the burden of understanding falls on the company, because it doesn't, but ethically there's a balance here.
Yeah. So the stem cells that are banked for adults are coming from adult tissues. Okay, so there's really only two types of stem cells that are readily found in adult tissues. The first are called hematopoietic stem cells, which are the stem cells that make blood. Red blood cells, white blood cells, platelets. That's what they do. And then the other type of stem cell are mesenchymal stem cells, MSCs for short. MSCs do everything else that the hematopoietic stem cells don't do. Okay, they have tri-lineage differentiation, which means that they can differentiate into all three layers, so the adipose, cartilage, and bone. But they are known, depending on the environment, they can become pretty much anything in the body. Now, just from a usability standpoint, there's much more opportunity with MSCs because it's not just limited to blood. Most of us are concerned with our brains falling apart, our joints falling apart, our skin sagging. Like, these are things that we're all worried about, all the other tissue types in our bodies. So if the cells that can regenerate them are mesenchymal stem cells, then those are the ones that you want to bank. So stem cell type is very important. Which type are you wanting to bank? Now, going back to the cord blood world, cord blood, when they are accusing companies of not disclosing what was being banked, or patients not fully understanding what they were banking, it's because they're told that, well, there's this potential that these cells can help all these diseases. Well, if the cells that are being banked are hematopoietic stem cells, then no, there is no direct correlation to any of these diseases that it could help. So it was not just them theoretically saying what could happen in the future, but it was just scientifically flawed and fraudulent, in my opinion. So, what are stem cells? What stem cells do you want to bank? Type first, then source. Okay. People get confused with stem cells and source. Source is the actual tissue. Where do you find the stem cells? Like, what source are you going to use to extract your stem cells from? So sources, popular sources: fat, bone marrow. Bone marrow has been used for a very long time. The latest one, dental pulp. Skin, people used to take skin as well. Skin is really not fun. Nobody likes to give a chunk of their skin. But dental pulp, I recently saw my dentist was doing that too. So it's getting around. Apparently they can isolate from wisdom teeth that are being pulled out, or even kids' teeth. So let's talk about the different sources. Let's talk about what these sources are. Again, it depends on the goal. What's your goal? Are you trying to bank hematopoietic stem cells or mesenchymal stem cells? For the sake of this conversation, we'll stick to MSCs. I think that's the most popular cell people want to bank, and it's a great idea. If you can bank MSCs, I'd say great thing to do, and they will probably be very useful. So when it comes to withdrawing these cells later on, we suspect that if MSCs take the position that we think they are going to, just given their potential, then they will become a very important medical tool for us to utilize in the future. Therefore, we have to be able to withdraw them when we need them.
Well, and when it comes to the sourcing conversation, I mean, there's scientific rationale for everything, but what hits me on a deeply human level is, of all those tissue sources, the one that it's easiest to part with is fat.
We have no shortage of fat in America, Jen.
And we've never had anybody argue with, the tablespoon of fat that in our particular process is required. They usually go, "Oh, can you take more while
That's
you're
the
there?"
biggest complaint. Why don't you take more?
Yes. So, and truthfully, well-natured joking aside, that is part of the consideration is, especially if you're banking someone, if you would like to be able to bank someone who is already ill or already medically fragile in some way, which of these is the least invasive source with a comparably high potential of yielding the number of cells that you need, and so on and so forth. These are all considerations.
That's a great point, Jan. I didn't think about that as being part of the consideration, but I think that's very important. Ease of sourcing that material. I don't think you'd want to go pull out your teeth per se, unless one falls out is one thing. But yeah, it's ease of source collection is a big one. But the quality of the cells that you can find in that tissue, especially if the objective is to bank, then we have to decide. And the decision would typically be the volume of cells you can find in that sample. I think that plays a big role, because the quality and the volume of cells that you find will directly impact, again, what you can withdraw in the future. So I'd say when it comes to sources, it's a number with stem cell treatments, stem cell banking as well, it's all a numbers game, okay? How many, if the objective is to bank MSCs, how many MSCs will you find in a sample of, let's start with fat, since fat is, again, readily available, easily taken out. Sounds great, checks off those boxes, but it's fat. Come on, it's just like, what could possibly be in fat? Well, fat turns out to be an excellent source for MSCs. Why? Because your MSCs in your fat are doing a great job keeping your fat exactly where it is. They are responsible for revascularizing that fat and keeping that fat nice and healthy. And we all know how hard it is to get rid of those pesky cells. So MSCs do a fantastic job. They're lodged in there. They're safe from the environment, if you think about it. They're not in circulation, so they're not affected by you getting the flu or anything. They're lodged in your fat, a very protected tissue source. And from 10 grams of fat, which is the amount we take here, 10 grams is under a tablespoon. Under a tablespoon of fat, you'll find approximately, on average, 100,000 healthy MSCs. 100,000. Okay, so that's a benchmark. In bone marrow aspirate, if you were to go get bone marrow taken, which is not a fun process, but there are physicians out there that say bone marrow is a better source. We have not seen that to be the case, especially when it comes to MSCs. Bone marrow, which is closely tied to, because all the cells that are made in your bone marrow, that's actually the hub where all your stem cells are made, is in your bone marrow. And then those cells get deployed into the blood system. Your bone marrow is highly sensitive to the environment, so it will behave according to, it can behave differently according to outside factors. So it is highly variable, but on average, you'll find 1 MSC per 10,000 to 100,000 bone marrow cells.
It doesn't strike me as a great yield rate.
Yeah, so the yield is not great. And again, because of the sensitivity of the source, the older you are, you've heard of this. I'm sure many of you have heard of this, where some talking head will say, oh, well, older people's stem cells are not as good as younger people. I think that is a common, it's not a misconception because there is scientific truth behind that, but it depends on many factors. It depends on the source,
but
right?
We get asked
on the
that
source.
a lot, though. We get asked that, especially for people who want to bank their cells to later participate in clinical trials. If they're older, they go, are my cells even any good?
Right.
Like, yes, depending.
Which makes so much sense.
Yes.
But if you told me, if you gave me a bone marrow sample, and I could definitely tell the difference between an 80-year-old and a 20-year-old. But fat is not always that case. Fat is very much similar, person to person. Again, at the beginning is when you can tell a lot of these differences. After processing and growing cells, you're growing the number, so now those differences become less visible or less noticeable as you go through the process. But at the beginning, yes, you can generally see these differences, and more so in different tissue types like bone marrow. Now, in dental pulp, you can expect 1,000 to 10,000 MSCs per tooth. That's very, very small amount.
Well, you'd have to pull a whole mouth of teeth to get
a comparable, a whole mouthful. Yes. I'd say it wouldn't be worth pulling out your teeth to do that. Certainly not enough to bank a tooth to, again, what are you going to be able to withdraw in the future, right, from that one tooth? From 1,000 cells, and Jan, we are experts in being able to grow stem cells. We can grow stem cells out of nothing if we need to. I mean, we are very good at what we do, but we still require a very healthy population of cells. And even a few cells, we can get them to where we need to go. But what you bank at the beginning will define that success. This is why you can't depend on source material alone. So when I say 100,000 cells per 10 grams of fat, you would say, oh, wow, Donna, that sounds like a lot of cells. That should be good to bank. No, it isn't. It's great source to start, but it actually isn't enough to bank either. In order to ensure that someone has cells left to withdraw in the future, you actually have to expand these cells. And we'll go into a little bit about what expansion means. But that's the number, okay? So the numbers are important for you to know. Now let's talk about what types of adult stem cell banking there are. So I tried to search companies, ads, services that people are marketing online for adult stem cell banking services. And generally they fall into 3 categories. We'll bank tissue for you. So come to us, we're gonna take your fat and we're gonna freeze that fat for you. Because fat contains stem cells, okay? The second is stem cell banking services that will bank, in the case of fat, stromal vascular fraction, which is essentially the tissue that's been digested and the cells are centrifuged and you get a pellet of cells. That pellet is a combination of cells. There's all kinds of cells in there, which they claim are all good, but the MSCs are in there and other cell sources are in there. And so it's what we call minimally manipulated or minimally processed material, cell material, that is stored. Again, if you're depending on the amount of fat or tissue that was taken, that initial processed cell or separated cell population is going to be dependent on however much tissue was taken. Yeah. So you can actually do the math on that. And then the third is culture expanded. So banking services that will take the tissue, isolate the cells, and then grow them in their number to expand the number of cells to store those for future use, because you will need large numbers of cells. Yeah. So those are the three types.
Well, and I think, it doesn't require a scientific degree or experience in the life sciences to just think organically about this and start to draw some conclusions about what problems may be in the industry. Starting with, as we've talked about, sourcing what you're banking, how you're banking it. And I think one of the major we've already touched on is, claims about future treatment. I mean, that's what resulted in the lawsuits that started this conversation today, largely. What are realistic claims about what banking anything might make available to you in the future?
Well, first of all, the stem cell field is growing exponentially, and the use of stem cells and the stories we're hearing and articles that are out there are just showing more and more use cases, clinical trial results showing great results with stem cells. And the problem is we're getting into the bad habit of mixing all that together, saying that there's promise in stem cells, therefore you should bank. Yes, that's not good. Yeah, that is misleading, because the stem cells that are banked, again, or the process by which those cells are banked is different depending on the lab that was doing that clinical trial or that test. So it's really, the results are exclusive to that, unless you can prove that the cells that were used are indeed the same, that they were processed the same way as said bank that is trying to sell you on the service. So if a company is saying these stem cells that you're banking are going to be good for these diseases, and they're using references from other clinical trials or other people's work, I would be highly cautious about that. Just because MSCs worked for X disease does not mean that the MSCs they claim they're banking for you are going to provide that same kind of result in the future. Yeah. Two totally different things. And I do think that the processing of cells is directly correlated to the efficacy of a cell. It's not the cell itself, not anymore at least. We've been doing MSC research for the last 30 years. I'm talking about the industry. And we see time and time again, when these cells go into clinical trials, it's a mixed bag of results. So that there means there's high variability. So just because cells prove that they work in a lab does not mean that they will prove to work in clinic, unless they are proven to work. They have gone through the clinical trial testing and all of that to show that there is consistent efficacy in a group.
Yeah, these are all really great points. And when it comes to banking for future use, also, I think there's a lot of misleading fear-based marketing out there, because it's one thing if you're selecting a banking venue, you're selecting a banking type, what we should be thinking about is what has the greatest probability or the most likelihood of being useful and being accessible to me if and when I should need it for fill in the blank. But the fill in the blank is actually a great mystery. I mean, if, heaven forbid, you have a multi-system illness or a complex medical condition, or there may not ever be a body of clinical research that's specific to that, your exact healthcare profile at the time. And so when I see all this marketing that's so focused on specific conditions or diseases, number one, I'd argue it's just outright unethical, given what you've said. But number two, I think it pushes people who are already concerned about their medical future toward a poorly framed way of thinking about what to bank and how. Because the future is ultimately unknown.
Yeah. Well, that's a good point. I think, and it'll be impossible, right? There will be never, we won't get to a point where we'll have a body of data for every living disease out there, right? Yes. But I think there are common denominators that we can look at. For instance, we study a lot of inflammation, right? Because chronic degenerative disease underlying problem is inflammation across the board. There's systemic inflammation. We study the brain a lot. Well, sure enough, in these chronic degenerative neurological diseases, what do we see? Neuroinflammation. It's consistent. So if inflammation is a problem and these cells are great at fighting inflammation, then theoretically they should be used for all of these indications, not look at them separately, right? So there you have it, right? It's not, this is a treatment or a cure for a specific disease, but really the mechanism. How are these cells, how have you proven that these cells keep? If MSCs in your body are inherently anti-inflammatory, then have you proven that after you process these cells and bank them for me, that they're still anti-inflammatory? Have you done that work, right? These are very important questions because it's really about the mechanism. So yes, I fully understand that there will likely not be a body of data for every single disease out there. But again, if you go back to the mechanism or the fundamental problem underlying issue of a disease that you want to resolve, like inflammation, then look at the mechanism of the cell. What cell can do this? Well, most roads lead you to an MSC, okay? But again, MSCs can only be useful if processed correctly.
And let's talk about the processing, because that's one of the, whenever we give tours here, that's my favorite part, because I mean, first of all, it looks really, the labs and everything are really cool. But when we talk about quality assurance, you see so many light bulbs. And this is what happened for me as well. The first time I came here is you walk through and you go, wow, I never thought of that. Like, I never thought of how are we ensuring that this product, or how do we ensure that any product, I mean, it's a whole new world of thinking about what we are purchasing. So how are we ensuring, and what should people be thinking about when it comes to viability of treatments?
Well, I love that when the word viability comes up, you're right. At the beginning, we took a lot of pride that our cells are like really viable, like they really are. And then it suddenly became apparent that, well, everybody has viable cells, or so they say. And so that is when we embarked on a lot of testing of comparing, like what really is viability? Because the viability threshold is actually very low. Most cells will read being viable. A good example is like freezing cells. When you freeze cells and you freeze them in appropriate methods, there are good ways of freezing cells. And in the banking conversation, you'll have banks tell you, we're the best at cryopreserving tissues and cells. Like, as soon as we isolate these cells, we're going to freeze them for you, and they'll be great. How do you know? Oh, because we know they're viable. Okay, well, what does that mean exactly? So the viability threshold is actually very low, because the tests that we use will show whether a cell is viable or not. It won't necessarily tell you, unless you do some other extensive, like proteomic analysis and some other analyses, to determine like how functional is the cell. Being functional and viable are two different things, right? Because you can be viable but not useful. Do you know any people like that? right?
To like control my face as you say that. Yeah.
Right. Someone can say we have 100% viable product, but a bunch of useless cells, right? That is actually the biggest problem in the development of cellular therapeutics, where cells are being administered, where a great majority of these cells are not useful in any way, shape, or form. But they're viable, so they claim. So when we started this, we would say, when we said they're viable, we meant it. Like they're really viable. They're happy, active. They're the most active of the bunch. They've survived the cryopreservation process. We've reseeded, replated them, and they've grown like, they've done a bang up job growing, and that's how we define the health of the cells. And we also look at what they secrete. Do they do what they are supposed to do? MSCs go into your body and they will localize to areas of injury, and they will begin secreting like a drug factory. They'll start secreting all kinds of factors, but it's key that they're healthy enough to do that, right? Are they potent enough to do that? That goes back to potency conversations. But when we talk about viability, it's important that we talk about functionality and viability as one, because all of that leads to potency, right?
Well, and I think too, part of the viability conversation and the potency conversation has to be around testing and how often and when products are tested. And that's a huge part of being able to look somebody in the eye and say, hey, I know what you're getting. Okay. I know what these thresholds and what these levels are. We can quantify this. We've tested it. And we see, unfortunately, too often in the industry, things simply being defrosted, for lack of a better word. And as anybody knows, if you've ever tried to freeze a bunch of stuff in your freezer, right, sometimes it goes well, sometimes it doesn't. So the mechanisms behind how a company stores these cells, I just can't emphasize that enough, because, again, it was such a surprise to me the first time I saw it, because I just didn't know to think about it. I didn't know to think about it. Even if you're looking at the research, you should, we still want people to be asking these questions about how things are stored, and really scrutinizing, do these procedures even make sense? Just
right.
From a layman's perspective, not even a scientific one.
Right. Yeah. So makes sense. Let's one thing is dose. I think dose is a great conversation, because it's one of those things that we don't think about, but if a treatment is going to be made in the future, we need to know what that order of magnitude is to determine whether banking will make sense. This goes back to the cord blood world, right? If an adult is going to need cord blood as a treatment, and remember, the type of stem cell in cord blood is hematopoietic stem cell. So let's say somebody has leukemia and wants to be treated, an adult. Well, how many, and let's say their parents, an adult whose parents had banked them when they were born, their cord blood, is that unit of cord blood going to contain enough cells to be useful? No, the answer is no. Not an adult, at least not an adult dose, maybe a child's dose, but and then also you do have other issues that if that was the indication, actually they wouldn't be able to use their own cord blood anyway, because their own cord blood is you would have to find a donor for that. So defeats the purpose of banking altogether. But you do want to think about the numbers. So in treatments, and this is where you can take other trials and kind of look at, well, what have other trials needed to use? Like, what have other trials used in their dose formulations for different conditions? And if you look at the doses these days, they run anywhere from 100 million onward. I think, we at Hope Bio, we have standardized dosing at 200 million cells, but we came up with that number with many years of testing to determine what is the appropriate safe dose. And so when it comes to banking, how many cells are you banking at the beginning so that you can achieve these sorts of numbers at the end? If the goal is to create treatments and perhaps subsequent treatments. Again, when you discuss the different diseases or claims that they make when banks tell you, oh, these cells will be good for Alzheimer's. Well, did they tell you that Alzheimer's will be, if you do plan on treating Alzheimer's with these cells, it will be a treatment that will be required for the rest of one's life? Okay. That's a truth bomb that no one really wants to talk about, but that is the truth. Degeneration can be fought by regeneration, but that regeneration needs to be continued because the degeneration is also continuing. So you will almost have to regenerate quicker than you're degenerating to make you, to at least stabilize the condition. And this is all potential, right? This is all treatments that will be in the future. But again, you have to be able to repeat. And therefore, the creation of, let's say, in this case, 200 million cells would have to be repeated over and over and over again, which would be billions of cells over the course of many years. Yeah. So can that be made from a single bank? That's the question.
Depends on what you bank and how.
Right. Depends on what you bank. But these are good questions to be thinking about and to ask. And the other thing that I think is not readily considered is most stem cell banks are banking tissues, okay, tissues or blood. When they do that, it's not under GMP regulations because most of the time when you bank tissues, you're banking tissues to use the tissue later on. You're not banking to do something further with those cells. I mean, it's possible. I guess it's possible, but the regulations that the bank needs to abide by are not, in this case, GMP regulations, which are good manufacturing practices that are in place for drug manufacturing. Because when you're banking a tissue, you're not exactly banking a drug. Those are different. Yeah, so FDA oversight is very different when it comes to drug manufacturing and tissue processing. They're two completely different worlds. Tissue processing involves, is the tissue collected properly? Is it processed properly? Is it stored according to has the donor received the proper testing? That's really the extent for GTP, which is good tissue practice regulations. Yeah, which is what most banks follow.
Well, and GMP is so much more comprehensive. I mean, it's everything from your facility to your processes to your people to your, it's everything.
Because those GMP regulations lay out all the steps and everything that needs to be done in order to make a drug. So where does the drug making process start? One can argue that it starts after banking. Okay, banking is not part of that process, that it starts after cells are banked and then we begin by thawing out cells that are banked and then processing for treatment. But no, most of the time, what is banked is what is being used. Okay, so that's why the GMP regulations are important all the way through. The cells that we bank here at Hope Bio are the cells that we will go on to process for treatment. Therefore, the whole banking process is part of the drug manufacturing process. That's why it is regulated under GMP. Yeah,
it's a big deal, and it's something that people should look for and often. And again, I know I didn't know to look for until the first time I came here.
So you and I are banked.
We are.
Yes. My whole family is banked, and every employee at Hope Biosciences gets banked at their 1-year anniversary.
I have to tell you, I still think it's the coolest employee benefit I've ever heard of. I mean, who says that?
You don't have to pay banking dues so long as you work here. Yeah. It's a great gift. I will have to tell you that it is a different kind of peace of mind.
Yes.
Now, when you think about peace of mind, it goes back to this concept of biological insurance. And I don't, as much as I don't like those words, it is, in a sense, it is, but it really should be viewed as insurance almost connotates a just-in-case scenario. Just in case of something bad happening, here's your insurance policy. For us, we look at this completely differently. This is not a just in case something's bad, here are your cells. We view the use of these cells to be a regular tool we have in our arsenal for the rest of our lives. The idea of banking, stem cell banking, should be a repository where your cells are stored so that you can withdraw them as you need them. And that need can be not just to treat disease, but should be also to prevent disease. And that is coming. In order to prove that, we have to prove that they're useful to treat disease. I think we are well underway doing that in many different applications. But I think the exciting work is really figuring out, well, what can these cells do for us if we do this for prevention? Or it's not even prevention, it's really early stage of disease. Yes. Way before any diagnostic can detect it.
Yes.
Precisely.
Yes.
Yeah. So as a recap, I'm interested in stem cell banking. What are the questions I should be asking the banks?
Well, I think we've touched on a number of them here. You should be asking about the laboratory and the manufacturing setting itself. You should be asking about manufacturing processes and quality control testing, cell viability, cell isolation. And these also should be answers that are readily and easily explained and given to you. We have a problem in the cellular therapeutic industry with transparency. And it's like anything else. We had a saying when I was brought up professionally, if you can't explain it in a sentence or you can't summarize it in a page, you don't really know it, right? And that same theory applies. If it can't be explained to you, that might be an indication that this company isn't the best fit for you. What would you ask? I have a few more actually that I'm thinking that I wish people would ask more.
So I guess if it was me, and I'm thinking about how I would ask in order, I would ask first, what cell type are you banking? Okay, so if they say MSC, okay, how many of them are you banking? And what's your process? Once you store these cells, how long are they good for? They should be good indefinitely, but I'd like to know how long they've shown they're good for so far. And can they be expanded? Like, if you know the number of how many cells you've banked, can they be reproduced? Can they constantly be used? Because I don't want to give fat every time I want cells. I have to know that the bank can make these. And does the bank just store? Do they also make the product? Do they make the treatment? Those are two different things. If I store with you, how will I know that this will be able to make a treatment? Most of the time, they might say, oh, we just store. That's not our responsibility.
Well, but I think a valid question is how many treatments have been released? And for me, a caveat to that would be have been released for use in the United States from stem cell banks stored with this company, because it's and this is a larger conversation about what you think has value, but if you're understanding that often companies who store are not necessarily the same as manufacturing for treatment, there still is not inherently visibly a lot of point. If a company has never released a cell bank for treatment and they've been around for years and years and years, well, what credibility or proof do they have that they ever will? Or that they could?
Jan, you know how we started with this? Because we didn't start in stem cell banking. Because for me, it was, why would anybody bank their cells if there's no way to use them? So we didn't start off that way. We were well into research. We were doing other things, and then we were collecting banked tissues from other places. Other companies were asking us to make the treatments. So we thought, we can build a business out of being a lab that produces treatments for banks, like other storage companies that are storing this stuff. So they would send us material that they've stored, and we'd have a heck of a time trying to find a viable cell. Now, we need at least one viable cell to make a treatment, but we had very difficult time finding good cells. So I think that's when we realized that the banking process is just as important. It's, in fact, the most important step to determining whether you're going to have great, healthy cells later on. Yes. Okay, so the banking process is actually very important and should be done in the same procedures, the same level of, the same regulatory level as the manufacturing of a treatment. They should go hand in hand. So that's actually why we started doing it. And then it became, there's still no approved way to use this material yet. However, if you are interested in banking your own cells, then this is a good option, because we know up front, because we do all the hard work up front. It takes us about 2 months to actually create your stem cell master cell bank. That means that repository of cells that we store for you, that we're going to constantly withdraw from in the future, that is takes about 2 months for us to make. So you need hundreds of millions of cells to ensure that you're going to have enough to reproduce on demand. So from a single tablespoon of fat, we can manufacture over 1,000 treatments over the course of someone's lifetime. That kind of a question is important to ask when you're dealing with these banks. But again, most of the time, banks are not producing treatments.
But like
they're
in
simply
the scenario you illustrated, right? If you'd walked into these stem cell banks where you'd been searching for material and hadn't been able to use any of it, and they know that, and you ask them how many of your products have been released and resulted in successful manufacturing of treatments, and they'd said, nothing stored here has been useful in creating a treatment. Again, you don't need to have a life science background to go, that's probably not somewhere I want to invest a hope for my future and store my cell bank. I want to store my bank somewhere that has a proven track record of storing cells or in a way that they can later actually be used when I need them. Mm-hmm. So I think it's a powerful question that I wish people would ask more. And then one more before I forget it. I wish people would ask more about their privacy, about their bio-privacy. Especially, these days we're having all these huge conversations about technical capabilities and the internet and AI and all these things. People should be asking us how their own biological material, is being kept private and safe.
Oh, that's a major one, Jan. And I think nobody's really thinking about that right now generally, but that's a big one. I think we have to commit to, at Hope Biosciences, we are a fully U.S.-based company. We have zero foreign investment. We are your information and your cells are safe in these four walls, and that's it. That's what we commit to, and we don't use your cells for other uses. If you pass away during the time you're banked, you tell us up front how you want us to handle your cells. You can transfer that custody over to us, or you can decide to destroy them. That's up to you. These are transparent, open. We have seen in my opinion, complete degradation of integrity when it comes to the safekeeping of people's genetic information, material. It's a big deal. So, I can't speak for everyone else, but I think it's very important, and you should ask those questions. I can tell you what we do here, and that'll be an ongoing conversation. I bet there are a lot of things going on behind the scenes. Yeah. Tissues and stored material is becoming more and more valuable. Do you know that the most valuable cells are pre-COVID cells? We have one of the largest repository of cells, right? Because of the sheer volume of people that bank here. And when we look at it from a value standpoint, the whole world has been exposed to COVID, as an example. And so cells that were banked prior to COVID have suddenly become very, very valuable, right?
Well, that's an interesting conversation.
Yeah. So what does that mean? There's a lot of things happening. We know this. We know those kinds of implications. Yeah.
Yeah, there's a lot to consider, and these are questions people should be asking, and these are questions that people can reasonably expect a company to answer, right?
Hopefully,
which again is how you can evaluate whether this is a place where you can put your trust, which is ultimately what you're doing when you bank your cells, is you're placing your trust. And so my hope is that this conversation will empower people to do so in a much more deliberate, conscientious manner, because when you do it that way, it results in, in what we offer here, which is peace of mind. And we both personally experienced that. And so
guaranteed.
Guaranteed.
Like if we get your tissue and we cannot find good cells and we can't successfully complete the banking process, you get every penny back. Even if we've spent, there was a time where we spent, 16 weeks on someone's cells and we couldn't do it? All that is refunded back to you. It doesn't matter. If we can't do it, we're not going to charge you a penny. That's how it works. Yeah. We know upfront whether we can do it or not. Yeah.
Yes.
So that is so should you bank your cells?
Yes, I believe you should.
Yeah. If anything, and this might be the real insurance of the conversation. If something bad does happen, what are your options to access your cells? Now, in the United States, the right to try law does apply for any life-threatening situation. It does apply to products that are under investigation and that have passed a phase 1 clinical trial. So our product, meaning the product the treatment that we make here out of autologous cells, have met all of those requirements. So therefore, if you are banked here, let's say, and something catastrophic happened, under the right to try law, you do have access to your cells. Now that's a big deal because in the US, we didn't really have an option beforehand, but now we do. So that is it worth it just for that? I'd say yes. I'd say based on a lot of data, I think people with high risk occupations, athletes, there is good reason to have your cells banked. Absolutely. Yeah.
Or, even complex medical situations where the conventional system perhaps isn't yielding the results, or you don't have access to the tools within that system in an affordable, truly accessible way. I think, stem cell banking can be part of the answer there, and a very large part.
Yeah. And if you're accident prone. Yes.
Not naming names, but
it's a good thing to do.
Us, yeah.
It's a good thing to do. Yeah. Yeah. Well,
I think this was a good one.
Let's talk about cost before we end.
Okay.
Since it's a follow-up to last episode.
Yep.
Last episode, we talked about the costs of stem cell treatments. Now, when it comes to banking, what is a reasonable cost you should be paying for stem cell banking? So it depends on the service. You put a price tag on it. If a bank is going to take your vat and stick it in a freezer, then you determine, well, how much value is that? would you pay thousands of dollars for that service? If there is more involved in the banking process, chances are the costs will get higher. So that's how you'll see the pricing work. A lot of times banks are charging sort of like subscription fees because there is a cost to keeping your cells in storage. It's no different from, banking your eggs. It's like the same thing. There are cryo tanks that have to be maintained and all of that. So you want to do the math accordingly. What are the storage fees? That's what you should be asking, because there should be constant storage fees. And what are the upfront fees, and what are the services that go along with that? Yeah. So, Well, we can tell you what we charge. We haven't done research on what other people charge, but we charge $3,950 to bank. That includes the cost of the a little lipo. That includes the cost.
Mini lipo, yes.
At least in our network here in Houston. If you are going out of state and we need to find a different doctor for you, that will be out of pocket. That will be additional out of pocket. But if you're ever in Houston, the World Cup is coming. If you are in town, it's a good time to bank. Come and bank. Slots are filling up very quickly, but please consider us for your banking needs, and we'll be happy to arrange all that for you. But we do that. We also have lifetime banking options. A lot of complaints you hear about people who have banked at birth, in particular, ongoing fees. A child is going to become an adult in a blink of an eye, and then the fees keep piling on year after year. And there is no limit to what they can charge. So it just keeps going up. Year after year, fees go up. So if you want to limit that, maybe there are lump sum options that you can pay to preserve those cells. And
Well, and on this note too, I mentioned earlier that to me personally, a question of how many treatments have been released in the U.S. matters, because if companies don't have a track record of manufacturing domestically, in our terms, because we're a U.S.-based company, that means you also have to travel if you plan to ever access your cells. And all of these are things that are to be considered at the time of banking, right? You should be thinking about this at the time of banking, not when you're panicked and hurt or sick and scared, and then finding out whether you can or cannot access your cells.
Jan, to even go down that road and all the questions that you would have to ask to ensure like proper transport, validation of those transport services and all of, it just goes on and on and
goes
on.
on and on.
Yeah. I think if I could give a recommendation, it would be to bank where the treatment is being made. So the less hands that transfer the material, the better, right? Yes. But that's just my...
And then you have peace of mind.
And then you have peace of mind.
You do. You have peace of mind because these things have been considered, these questions have been answered, and then whatever your future holds, you feel that you've been diligently prepared.
Yeah. So they're not just stored cells. Okay, that's the wrong way of thinking. They are treatments that you'll use, that you'll want to use, not you might use just in case something bad happens, but it's treatments that we all want to use. I would want to use my cells as opposed to some of the options that are available today. I think we can do better.
Yes.
Yes.
Yes, I know we can.
Yes.
And I can't wait for the day when it's truly accessible as prevention, because that is going to be a game changer.
For sure.
Yeah.
Okay. Well, until next time. Thanks, Jan.
Thank you, Donna.